Hi, the following content is for educational purposes only and does not constitute medical advice. If you are a healthcare provider, please consult with your affiliated institution or local ethics committee for ethical guidance related to clinical practice.
Hello, BioE 131. This is module 31 on genetic engineering. David Magnus.
Let's start by just talking about some of the risks and benefits of genetic engineering technology. And as we're talking about them, let's think about what those technologies include in the broad category of genetic engineering. Sometimes these are called genetically modified organisms, GMOs. The technologies include traditional gene transfer, for example, using viral vectors. Often in non-human organisms, they use essentially gene guns to shoot genes into things.
That's mostly for plants, right?
That mostly for plants. It now includes CRISPR as a much better, more efficient, and more powerful set of technologies, and that includes the possibility of gene drives, which you've heard about in your technical briefing. What are the possible uses of this? Where are the areas of application? So genetic engineering of plant crops has been around for a long time, including food crops, feed crops like corn that's used for cows, cotton, more exotic areas like something called PMPs or plant-made pharmaceuticals. Those are, the idea would be to genetically engineer corn so that it produces human monoclonal antibodies, and so you can get a cheaper, more efficient way of developing biologics or very expensive pharmaceuticals. Genetic engineered trees, believe it or not, for the paper industry. There's a lot of work going on in that space. There's some genetically engineered grasses, genetically engineered animals. Again, for food, there's something called the super salmon, which is a genetically engineered salmon that grows large much more rapidly. And sometimes there's genetic engineering of fish for fun. There's something called the glowfish, which we'll be talking about in a little while, where you take fluorescent genes from one species and put them into other species. That's been done for glowfish. It's been done as an art installation. It was done for a bunny rabbit that glows in the dark. There's genetic engineering of yeast for the wine industry. There's several commercialized strains of GE yeast for wine. And of course, gene drives have been developed to eliminate pesky mosquito variants that carry disease, which we'll be talking about more later. So what are the potential benefits of all of this? You can create plants that are disease resistant. You can genetically engineer things so that you can use less pesticides and herbicides, therefore it could be potentially better for the environment. You can get improved yields. You can get enhanced health benefits. You could grow food in new regions that you couldn't otherwise. You could have cheaper capitalizations for biotech pharmaceuticals to get more pharmaceuticals. And then for the gene drives and mosquitoes, there are 4 primary targets, but you could wind up helping to eradicate the carriers of serious disease pathogens in human populations. So those are all pretty powerful benefits of the technology. What are the risks that are discussed in the literature? So there are unexpected allergies that people are worried about. DuPont didn't commercialize, but DuPont was doing research on a version of soy that had some genes from Brazil nuts. And then they discovered that the proteins that were produced by the genes that were being put into the soy would produce nut allergies in people who are allergic to nuts. They did not commercialize that product, but people worry about that. It's one of the things that's challenging for these are all parts of the broader concern of food safety. So the really big heading here is food safety. People are worried about the fact that it's challenging to segregate the food supply system. So for example, there was a version of BT corn, and Russ will have talked to you a little bit about BT corn or soy BT products in your technical briefing. So Bacillus thuringiensis, which is essentially a biological pesticide that's commonly used, but there are different versions of BT that produce different proteins. One of those proteins, something called crinine C, was not approved for human consumption because they were worried it would turn out to be an allergen. And then people were doing tests on Taco Bell taco shells that were sold in the supermarket. They found quinine C, which was not approved for human consumption, in the food supply system. It cost the company that produced that a huge amount of money, as they had to go through and do testing to have that taken out of the food supply since it was not approved for human consumption. There's no evidence that anybody had a serious adverse negative reaction to it, or an allergic reaction. But again, it highlights that even when we have things that we think are not safe for human consumption, it's very hard sometimes to segregate them out when they're being grown for animal feed or other kinds of non-human consumption purposes. Same kind of thing happened with some plant-made pharmaceuticals that wound up getting accidentally into fields in succeeding seasons that were not having PMP, and not only just corn, but also soy. And 155 acres of conventional corn wound up being burned, and 500,000 bushels of soybeans were contaminated by plant-made pharmaceuticals that wound up in those products. And you hope that the USDA APHIS inspectors caught all of them, but some people think that's evidence of the system working, and others think it's evidence that there's actually a big problem. Maybe even bigger. In general, our food safety system, we think, works reasonably well for GM. A lot of people are worried about environmental impacts, another set of risks. There could be escape and create a weed problem. When you develop hardier versions of plants, you could wind up creating them and turning them into weeds. You could get outcrossing, in which other kinds of plants could become super weeds as a result of those genes being spread into other organisms. You could even have horizontal gene transfer, where bacteria could be moving genes from one, or viruses potentially could be moving genes from one plant to another. Can viruses do, actually, I don't know the answer to that. Can viruses do horizontal gene transfer, or only bacteria?
I guess in principle they could, because if they acquire a bacterial fragment of DNA, and if then they increase their range of infectivity. But by far the most common would be the bacteria who actually have explicit machinery for injecting their DNA into other bacteria, which is quite amazing, and leads to bacteria sharing genetic tricks much more quickly than would happen by natural selection without that mechanism.
Very interesting. So those are all ways in which genes could be spread throughout the population. And then people also worry about the impacts of those in terms of things growing in environments they're not supposed to, ecological harm, ecological damage, the loss of wild types. There's a lot of species of maize in Mexico that were thought of as land races that have been contaminated by more modern versions of corn that have been found in those populations. And then, of course, there's some debates about potential impact on non-target populations. You're designing something to be a pesticide for one organism, but maybe it will be also wiping out other things. There were concerns about monarch butterflies being killed at one time. Not clear if that was really a result of those GMOs, but those are certainly a concern. There are different solutions. Just to give an example of a set of solutions, like management of risks for plant-made pharmaceutical corn, you can try and find ways of creating containment systems through things like genetic use restriction technologies. You can do whole corn harvesting. You can do more careful monitoring.
How does whole corn harvesting help the problem?
Because one of the worries that you've got is that there's going to be the spread of tassels and to other fields, and so there are ways of essentially
So you just take everything out of the field so that there's no biomass left to cause trouble. Okay.
And you do it in such a way that you're less likely to have spread.
So it's the whole, yeah.
Yeah. So you take the whole plant rather than just the corn off.
Yep.
If you take just the corn off, you're likely to spread. When you take the whole plant, you don't get that as much. There's physical confinement methods, there's temporal confinement methods, there's bioconfinement methods, monitoring and inspections. There are all kinds of ways of trying to mitigate those risks. But those are risks, potential risks, and then there's debate about how risky they are. There's potential social and economic, in addition, so we've talked about food safety and environmental risks. There are also social and economic impacts, risks to organic markets due to contamination. European markets response is important to note. That's a big impact when when USAID has wanted to send GMO food products or even seeds to less developed countries. The challenging thing there is that they may wipe out a lot of their economic markets because they were selling in Europe and Europe won't buy some of those GE products. So there's a recognition that there are some markets that don't accept GE products, and that could have a difference. There could be impacts of higher yields economically and make a big difference. There could be intellectual property rights. And I've actually written some stuff about biopiracy and how this can have negative impacts on less developed countries. So those are all the risks. And we've already talked about the benefits, and you know about utilitarianism. For utilitarianism, they would be weighing all these risks against all these benefits and make a decision whether you can mitigate the risks and make the benefits large enough to make it worthwhile in different cases and different technology applications, or whether or not the risks will outweigh the benefits in different technology developments and decide whether it's appropriate. And that's it for this video.
Thank you for attending this video. We'll see you in the next one.